one paper of mine tried to and managed to replicate this disease-associated state in a dish in vitro by specific compounds that we expose the cells to, and then they really drive their state into this disease-associated phenotype. So now we are able to also functionally study these cells because we don't really know yet whether they are beneficial or detrimental to disease, and also depends on the state again.
Hello everyone and welcome back to the Neuroscience and Beyond podcast. I'm your host Willem, and together with my amazing team of graduate students, we're on a mission to make science, especially neuroscience, more accessible and exciting for everyone. Today, we have the pleasure of hosting Dr. Verena Haage. After a successful postdoc at Columbia University in New York, Dr. Haage is now a guest scientist at Charité Berlin, one of the biggest university hospitals in Europe. Her research focuses on microglia, the immune cells of our brain. These tiny yet mighty cells act as the brain's cleanup crew, constantly patrolling to remove debris and maintain neural health. Beyond their housekeeping duties, microglia play a pivotal role in brain development and are increasingly recognized for their involvement in neurodegenerative diseases like Alzheimer's. Dr. Haage is also a co-founder of SciLead, an initiative dedicated to fostering sustainable leadership and improving research culture in science. So tune in to learn more about microglia and their crucial functions in both healthy and diseased brains, and the cutting-edge methods used to study them in animals and humans. We will also discuss the importance of leadership and mentorship in scientific research. If you enjoy the podcast, don't forget to subscribe and leave us a comment below. We love hearing from you. And now, dear listeners, please enjoy the conversation with Dr. Verena Haage. Hello everyone. Today I have the pleasure of hosting a friend of mine, Dr. Verena Haage. I will continue addressing her Verena because we know each other from before. Thank you for coming all the way from Berlin to our podcast.
Thank you. It's a pleasure to be here.
So we met, I was thinking earlier today, 6 years ago. I was writing my bachelor's thesis, and you were at the same lab, or I came to your lab actually in Berlin at Max Delbrück Center for molecular medicine in the lab of Professor Kettenmann, where the focus of research was glia. And you worked in particular with microglia. So these are very interesting cells. We're going to hear a little bit more or a lot more in this episode. And I'm very excited, not only because the topic is microglia, but also I'm very happy to have a friend who is now a successful scientist in our podcast.
Thank you very much.
So Verena, tell us about microglia. What is the focus of your research?
Yeah, the focus of my research has been shifting over the years because this year it's been 10 years that I worked in microglia research. In the beginning, it was very much focused on mouse microglia, physiology and development, and also in the context of glioblastoma. And over the past 4 years, I shifted to human microglia. So I worked mainly on diversity or heterogeneity of human microglia in different contexts, but rather related to aging and to neurodegeneration. There are some parts of my work, though, now that are also related to glioblastoma and microglia in this context.
So these are very, very exciting topics. I would suggest let us start by defining or saying a little bit more about what microglia are. So as we know in the brain, many people are familiar with neurons. They are the stars, so to say, in the brain, but we have also glia cells, and you have oligodendrocytes, astrocytes, and microglia. Tell us more about microglia.
Yeah, microglia are basically the resident immune cells in the brain. They are the brain resident macrophages and the first line of immune defense in the central nervous system. And yeah, for quite a while, they haven't been really looked at, but then they were discovered to be involved into many different diseases over the course of the last few decades. And microglia have basically this role already during development to ensure that synaptic connections are pruned in the right way so that we are wired in the right way, and then throughout life to maintain homeostasis in the central nervous system, in the brain. And they do that by maintaining the brain in balance, so to say. And if anything happens to the brain, such as stroke or glioma or ischemia or anything, any type of neuroinflammation, they are the first, so to say, line of defense that starts then to orchestrate an immune response.
So there are defenders in our brain, right?
Yeah.
I don't know how is the right term, like glia or glia. I never knew that, so I'm sorry if I tell it wrongly. I have to be ashamed that I was in Ketman's lab and I don't know how to pronounce this. But yeah, microglia, let's put it that way. You mentioned they were discovered a while ago, and the story is actually very interesting, right? This scientist Pio de Rodrigo Ortega, who actually spotted them, and this was together with the time where Cajal was working, right? And then he described the cells very accurately. Actually, this is more than 100 years ago. And at some point, people just left them away as something not important. And then again, in the 70s, 80s, scientists picked up and then started researching. And Ketman was one of the leading scientists, and you guys, all of the PhD students in his lab, were afterwards very successful in the field. You and other like Francesca, Alice, Amanda, I don't know her, but I know that she's really also in this field. So yeah, it was really excited to be in this lab and to learn about microglia. And this guy was actually Ketman, one of the people who collected all of these things and then spread it out to the world again. And it's fascinating. So you mentioned you worked with these cells in mice and in humans. Let's start with the mice. Do you see any difference of how microglia behave, let's say, in different brain regions, for example?
I specifically have not looked into that. I think single-cell RNA sequencing studies have shown that there are transcriptional differences that are region-specific, that are also, though, time or temporally different. So depending on in which state you are, and also the incidences that happen to a brain over the course of life, they look different. But in general, maybe to say a little bit more, there are differences between species. So human microglia differ, partially at least, from mouse microglia, which is quite important for medical and therapeutic approaches to study them. And there are also differences. There was a big landmark paper a few years ago, which also found that there are epigenetic differences between those cells from different species, which indicates different regulatory mechanisms, which of course is also important to know if you look into therapies and other approaches or signaling pathways. And there are also differences in general in gene expression between these two different species.
Mm-hmm. I know that you guys, with another very good scientist, Dilansu, you had this paper, you worked on this paper about difference in sex in microglia, right, in mice. Can you elaborate on that?
I can't really elaborate on all of the specifics, but it was one of the first. There was one earlier paper describing in mice that there are differences that are sex dependent in mice, and that they differ in transcriptomics, so on RNA basis, but also in proteomics, and that this is also specific to different brain regions. Mm-hmm. And I think that also, depending on therapeutic approaches that you then actually apply to mice, that they might differ in their responses to that, which has like a huge implication for any type of clinical trials and translational medicine, preclinical translational medicine.
Mm-hmm. This is very important because people who are not familiar with this medical testing field, a lot of the drugs that are being developed, they are tested in males predominantly because of different factors. And now by showing this, actually, that, hey, there are differences, and females are also important in this regard, not only in this, right, in many others, but it is crucial for any research that is developing drugs to also test their drugs in female and male mice, right? And yeah, I found this paper and this work of yours really, really fascinating, because this is just like, in a sense stating the obvious. Hey, there is a difference, but kind of nobody picked up this for a long time. I don't know, for whatever reason, and it's important that you guys did it, and it's very, very cool that the results show exactly things that you would thought of. Just to clarify something for people that are not familiar, what is transcriptomics and what is proteomics, for example?
Yeah, so one of these big dogmas that you learn when you study biology or genetics is that you have all our genetic information encoded in the DNA, and basically whenever this gets translated into functional units, which are the proteins, it goes through transcription into RNA bases, which is what we can capture with transcriptomics. So it's basically a transient photography or image of what's happening in a cell towards moving towards functional units of a cell. And with proteomics, we really capture what is actually happening on a functional basis right now in the cell.
And do you use other tools to study microglia, for example, or these are the primary things that you're doing?
Right now, during the postdoc, over the last 4 years, as a primary readout, we actually use single-cell RNA sequencing mostly.
Another very fancy technology.
Which is basically looking into transcriptomics, but on the single-cell level. So we have a much more focused approach and see on a single cell level what's happening within the brain or within each microglia in the brain.
Why is this important? So you can look, let's put things in perspective. So let's imagine our brain, let's say the hippocampus, right, where the memory is formed and etc., very important brain region, and you have microglia cells there as well, not only neurons and other glia cells, right? When you have now single cell RNA sequencing or SIC, what do you mean with single cell? You point one cell and in this particular cell what is being translated, transcribed, or you have a bunch of cells that you actually test or examine afterwards?
Yeah, so what happens there is basically, if we stay with this hippocampus example, we would take the hippocampus, we would prepare a single cell solution. So we would basically dissociate the tissue into a single cell solution, which can be done by different methods, and different other friends of ours have also shown which methods to use in order to preserve the transcriptome in the best way. So that would be manual dissociation on ice versus enzymatic digestion, which actually alters the transcriptome quite a bit. So this is not really something that is recommended at the moment in the field. With this single cell solution, you would then go into a single cell RNA sequencing workflow where you prepare actually from each single cell a library that then gets sequenced and then also computationally analyzed. So there are methods right now available where you can look into what you were saying, more spatially correlating transcriptomics with actually the position of each single cell more or less within a given brain region, for example. So these are very advanced methods where, for example, where you do spatial transcriptomics. There are several providers available to do that.
So in a sense, when you say single cells, it means like single cell type, right? Not one cell, but like all the glia that you dissociate from the tissue. Is that correct?
So what we would actually do is we would dissociate the hippocampus. We would isolate microglia. Yeah. So we have only microglia, and then we do transcriptomics on the single cell level of every microglia. So in the end, we actually zoom into the population of only microglia, and then we can really look into each single cell. Like imagine you zoom into the brain is Germany, you zoom into one city like Göttingen, and which would be the microglia, and then you zoom further into every person that walks on the streets, and you can assess so to say their characteristics, which then would be single person studies. So that's what we would do.
Okay, now I understand it fully, because always I hear single cell sequencing, and I'm like, okay, what exactly is this? I never bothered to read like really thoroughly, but now when I have expert and it's explained really, really nicely, I'm happy now that I know also what is single cell sequencing. And what kind of information does this give you, for example?
It actually gives you the information what every single cell that you look at and that you sequence is doing at the moment with their genetic information and with regards to translation into a functional unit, so to say protein. So you get a snapshot of what a cell is most likely up to. If we assume that transcriptomics kind of correlates with proteomics, so with that the amount of RNA correlates with the amount of protein, which is also not always true. But we can still try to at least assume or predict potential functions that might be happening in the cell based on what the cell is right now showing us in their transcriptome.
And in principle, you can, let's say, manipulate these cells or the whole organism. Let's say the mouse, you can expose it to a certain diet or more physical exercising or something else, and then you can again see, okay, is there any difference in this transcriptomics and proteomics? Do you do something in these lines, some experiments?
So you can definitely do this. I actually worked on this with regards to one specific project that I conducted over the past 4 years. This project was focused on finding a drug that potentially induces a specific subset of microglia. So maybe to go one step back, so a few years ago, or maybe 10 years ago or so, before single-cell RNA sequencing emerged, the microglia field thought or was talking about two different states mainly. And already 5 or 10 years ago, we were already saying that maybe this is not enough because these two states, M1 and M2, which were anti-inflammatory and pro-inflammatory, that they were right. We were thinking that they were capturing more or less the biggest phenotypes of microglia, but they were defined in a dish, so in vitro, by throwing LPS onto the cells. And this was a little bit artificial. So there was already this idea that there might have been some state in between that then actually makes the cells go into one or the other states. But now with single-cell RNA sequencing, we are able to actually capture the full spectrum of microglia states. And we are still trying to do so because there are now many different labs publishing many different states, and we need to actually find a common nomenclature in microglia research. But you could actually do that, treat a mouse, for example, which I did in one of my projects with a specific drug in that case, and then either from the whole brain or from specific hemispheres or specific brain regions, actually, that's what I meant, isolate microglia and then see how the drug actually affects microglia or also any other type of cell. You can also isolate neurons and see whether and how it changes specific subtypes of neurons. But before this technology of single-cell RNA-seq, we were not even aware that we didn't have the technology, so to say, to zoom in and see how many potential functional states there might be.
Let us also, as you mentioned, step back and talk about these two states of microglia because they're very interesting, the activated and the non-activated microglia. Can you describe them, for example?
I mean, this one state would be more anti-inflammatory, which is basically secreting a specific array of cytokines that are acting anti-inflammatory and which were thought to be rather potentially beneficial in certain contexts. The other state would be pro-inflammatory, which secretes then pro-inflammatory cytokines like TNF-alpha or interleukin-1 beta or TNF, I already said that, interferons and so on, which would actually feed more into this like active loop of inflammation and potentially also exacerbating specific or certain inflammatory conditions. Yeah, so these are the two.
Yeah, it was very interesting that people say like the active and non-active, but actually the non-active, they, check out their territory constantly. They're in contact with other cells and they're actually pretty active even when they're non-activated or whatever. And this can lead to some confusion for people who are not in the field. And yeah, these factors, these TNF, alpha and gamma or whatever, these are also play a role in glioblastoma, as long as I remember, and tumor formation, or I'm wrong? You don't know?
I'm not really an expert in cytokines in a tumor environment. So, yeah.
Yeah, I think there was this guy in the lab back then who was a medical doctor and still is, from Croatia. I don't know, Marko or something. Do you remember him? I think he worked with some stuff like this, but it doesn't matter. This is a different story.
I think also in glioblastoma you have different microglial states polarized towards different subsets, so to say, and there might be definitely one that expresses pro-inflammatory cytokines, and it might be actually feeding the tumor to progress.
Yeah, when we say cytokines, these are just specific molecules, that these particular cells release, and also other cells can release them. But if you want to know more about them, please read up. So you mentioned that human microglia and mouse microglia are different. You mentioned also there are some difference in epigenetics and transcriptomics, etc. But what is the most obvious difference? And I know that there are so many interesting studies testing drugs in mice, and what is happening with these drugs when we apply them to humans, for example?
We cannot always be sure that these drugs are directly translatable from preclinical studies into clinical trials. What is different between human and mouse microglia? I'm also not an expert. However, I know that specific marker genes for example, homeostatic microglial markers versus some activated markers are potentially lacking or different, or are just different homology there. And so it just really depends also, I think, which pathways you study. That's actually the major thing, that you are aware whether the pathway you want to look at, and if you have a drug in that sense, then you know where it works, hopefully. Sometimes we also just know what it does, not so much what it worked or where it works yet, but that we need to always check and be sure that these pathways can actually be translated, and these are pathways that we can look at in the mouse to verify, or to actually discover and then verify later in clinical trials, or first in human models.
So when we study mice or something in mice or in rats, for example, we have a lot of tools with which we can manipulate. For example, certain cell types can express some fluorescence molecules or some other markers to give us additional information. We can do this single cell sequencing, and we have basically all the tools that we want to study this particular cell or interaction with other cells. However, when we have humans, I mean, it's, I guess, difficult to get samples from humans, especially, samples that are still alive, that the cells are alive. What kind of methods do you use to study microglia in humans?
Yeah, it's a good question. And so there are different possibilities, but obviously they are limited. So the simplest way to do sort of screening experiments there are different human microglial cell lines. There's one that is quite famous, which is called HMC3, which is a cell line. Human embryonic cell line means HMC, and this was immortalized. So that's already the first kind of problem there, because they kind of grow like cancer cells. So they multiply very quickly, which is not really the nature of microglia.
Let me interrupt you. What do you mean by immortalized? Yeah. With this cancer behavior? Yeah, there was a vector transduced into these cells, which then leads to just unconventional, or just to them being multiplying very rapidly, like a cancer cell. Uncontrollable
growth, so to say, more or less.
I see.
So this is one system, and we use this in the very first line of experimental approaches or studies. And then the more fancy ways to work on microglia are, if we stay with cell lines, iPSC-Derived microglia. So there are a lot of different protocols out there, which have been, I think, published since 2017, like a few milestone papers, and most of the field works with one or the other of those, and also constantly tries to improve those. So there you differentiate from hematopoietic stem cells, induced pluripotent stem cells, and then you go along a trajectory where you feed specific signals such as cytokines and other growth factors and so on to these precursor cells, and then they differentiate into microglia. We also worked with these cell lines, which are quite close to, or right now are the closest model, or so to say the gold standard, but these model systems also have their flaws. There is a big problem also with sort of reproducibility or variability. Even if the same person differentiates on one day and then on the next day, the cells can look quite different. And so there's also a need in this field to look into gold standards with regards to how many lines to use and how to differentiate them and how many replicates and so on to do. Then there's more advanced systems where we use, not me, but we collaborated with labs that do that, cerebral organoids, which per se they don't have microglia intrinsically, because microglia come from a different layer in neurodevelopment or in general development, but you can actually implant microglia at a specific state into the cerebral organoids, and then they would integrate. There are some papers out there as well which show that this works quite well, and so we also collaborated with a group in one of our papers to look into this. And then you can in order to have access to human tissue, of course you have to establish the collaborations, which is quite difficult, which was perfectly set up in New York, and which was one of the reasons why I went to Columbia University to work there. And so there were, with a lot of different labs or clinics across the country, there were collaborations where we would get postmortem samples, which means people that donate their brains after death, that we get parts of their brain, and then we isolate cells and do whatever experiments we want to do with these cells. Of course, the quality of the cells depends on the postmortem interval, so the time of extracting the brain from the donor and the time it takes to travel to the lab. And so to have better quality of cells, we also collaborated with neurosurgical units, specifically one at Columbia where we got glioma tissue. So basically biopsy tissue, sometimes also from epilepsy patients where you have access to kind of fresh brain tissue, which the quality of cells is much better. And we then also focused some validation experiments on glioma, even though we were not necessarily mainly interested in that, but just because the quality of cells is just so much better.
I have now actually access to glioma samples, and I'm very happy finally after so long, such a long waiting. I have so many questions about everything that you said right now. So first one is, do you know if the brains that people donate, they are, I guess, frozen, right? Cryo-frozen or?
It depends very much so what you decide with your collaboration partner in the clinic. The ones we got, because we do single cell RNA sequencing, they were in media. There's a specific hibernate A media in which we get the brains delivered. Because we want to do single cell RNA-seq, we need the cells to be intact. If we would get frozen tissue, we could do single nuke RNA sequencing. But mostly, I mean, I know that there are some people trying to work on protocols, or actually also a friend is trying to work on a protocol where you can, from frozen tissue, isolate live microglia. And I think this protocol is just still, the paper's still being written up, but it's possible to do actually also these type of analyses like proteomics and single cell RNA-seq, but we are not, we don't have this protocol available. So for us, the tissue is not frozen.
That is very interesting. Hibernate A. Okay, I will remember that. So basically they put them into some state that they're static or something, but still kind of intact so that you can analyze them.
I think the media just has some specific factors that are keeping the brain tissue more intact. Yeah. And sometimes you can also add FCS, but that might, of course, also change a little bit the transcriptomics of your cells, because it's been shown that FCS alters transcription.
What is FCS?
It's fetal calf serum, which many people use for cell culture. I mean, most commonly used for cell culture, but also in microglia research, since it's been shown that it really affects transcriptomic states of microglia, there are certain protocols which try to leave it out and substitute it with different factors.
And in this this protocol that your friend is developing now silly question is coming. Can you, so cryofixated brains, can you then take the cells and then immortalize them in principle out of this? What do you think? Is this in theory possible, or?
I think it might be possible, yes. I mean, of course, then you alter again the transcriptome and everything, but it must be possible, I think.
That is very
but
interesting.
This also depends, of course, how easy, because fresh human microglia are very reactive, and it's difficult to transduce them with like a virus or something. I think people manage, but it's not an easy thing to do.
Very interesting, very interesting. And so the induced pluripotent stem cells, these are very fancy things that people, let's say, take a cell from a skin of a person and then reprogram it to something else that is, for example, microglia in this case. Have you worked with these cells, like with particular patient, for example, like somebody with a certain disease, or you don't have this in the lab?
We did not have that in the lab.
Yeah.
We are collaborating for that, but currently PhD student is setting this up. But it's definitely the way to go, and many people have established and are establishing kind of big libraries of these iPSC lines derived from different patients, from different sex backgrounds, different ethnicities as well, and
How do you think, is this like a good future technology for personalized medicine, for example?
Yes.
Yeah, it's definitely different than animal models, right? But still, it's like it's not capturing the complexity of a full organism, right? So this is one of the disadvantages. And if I continue in this line of thought, so these cells that you are having, for example, the iPSCs or the other ones, the cell lines, would you say that they capture mostly what is happening during early development, or can we take them as example for adult cells, so to say?
They look... There's a paper that compares that, actually. What I know from my work is where we looked at those on the transcriptomic level, but also on a sort of kind of proteomic single cell level with a method that we tailored for specifically to look into microglia with like a limited amount of the proteome on the cell surface, and in addition to combine it with the transcriptome on a single cell level. We saw that they are looking quite different to this HMC3 line, and they are also looking quite different to actually human freshly isolated microglia. So they all cluster in very specific areas if we do cluster them using a computational analysis. However, there's also a little bit of overlap with freshly isolated human microglia, which are from adult human and actually aged human patients isolated. So I would say they kind of recapitulated, but not fully recapitulated human microglia. And I think we need to perform actually more studies where we compare them across ages, where we compare them across disease backgrounds and so on. The problem is that it's very expensive to perform single-cell RNA sequencing. So a lot of labs would need to collaborate together and use the same workflow so that it's actually comparable.
Can you give us some idea, for example, for one tube of microglia, how much do you pay, or your institute? Do you know that?
I think we always calculated one sample which could be treated cells or from one patient or so on, for about $2,000 to $3,000.
This is one Eppendorf tube, or
that would be one little Eppendorf tube from like one source. But there are technologies where you can actually hash samples together. So there's like a workflow from 10x, which is one of the big companies that is doing single cell RNA sequencing, where you can label your samples. Imagine you have 4 samples, up to 4 samples. You can pre-incubate each of these samples with a specific antibody that has an oligonucleotide to it. And then instead of doing one library per cell, 2 libraries are performed, or 2 libraries are produced with different sizes, because the one from the antibody that is tagging a specific protein on the cell surface is much smaller. But then computationally, you can basically tag them, throw them all together, sequence, and then computationally you can differentiate them again. And then you can look at 4 different samples. Of course, you have less cells per sample available, because you give up like 3/4 of the potential cells that you can analyze in one for other samples. But that's a way to kind of save money.
It's crazy what people are doing. And for example, if you want to have like a full experiment with full number, like good statistics, you would do like this. You're not gonna give the sequencing facility or company one Eppendorf, but you would give them, let's say, 4 different experiments. Each would have like maybe 4 replicates. Am I right?
That would be the case for bulk RNA sequencing.
Yeah.
That's not necessarily the case for single-cell RNA sequencing. I think there are debates. Some biologists some biologists would say each cell is kind of an experiment. So they would say one sample with a sufficient number of cells that is being analyzed is enough. But then maybe you look into 10 or 20,000 cells per patient, not into maybe 3,000 or 4,000 cells. Of course, it's always better to run more, but in practice, we never ran 2 times single-cell RNA-seq from the same person. Then you would maybe look into different brain regions, or you would look into their blood cells and then compare them to the brain cells or something like that. But we would not really do that, actually.
So in the field is established.
What I did for mouse experiments is where I tested a drug on different mice. Or actually, I tested 3 different drugs on mice. And different mice, I mean on both sexes, because you cannot publish any study anymore thanks to many of those previous works without looking into both sexes. So there we had 4 mice per condition. So 4 mice for control and 4 mice, for example, for the drug treatment. And then we did single cell from each. But that's because there's also, of course, variability within even if it's inbred mice strains. So to account for that.
Yeah, of course. Reproducibility is important also, right? This is something that we can maybe touch upon a little bit later. And then how hard it is so this is a huge amount of data, right? How hard it is to make sense of all of this?
Yeah, I think it's very hard. I personally have not done or learned the analysis. So the beauty of the center where I did my postdoc is it's called Center for Translational and Computational Neuroimmunology. So there is a beautiful bridge, so to say, between computational and between wet lab neuroscientists. And the communication works quite well. It takes a while to actually communicate properly, like to communicate from the computational side to the experimental side what you want and the other way around. But yeah, to work on this amount of data, especially if you have multimodal data, like proteomics and transcriptomics from one experiment or something like that, it requires a lot of training. Yeah.
Yeah, it's basically professionals who are bioinformaticians or computational experts. I don't know. They know their things, and of course, science. When I looked in some papers from professors, I don't know, like that are directors now, and they're like early papers. Then you see like three figures. That one is EM, one is electrophysiology recording, and one is something, and then you have Nature paper. I don't want to disrespect any of the research performed in the past because it's awesome, but I want to say that nowadays, in order to publish in the big journals, you need to have basically every new fancy technology and perform so many experiments in different animals, in humans, etc. And then it's so crazy, and one person just cannot keep up with all the technology and all the information. So therefore, there are specialists, and it's good to be so, because if you're specialized in, let's say, bioinformatics and this kind of reading the information, then you're very good at it. And if I have to do this on my own, then, or you or whoever else, then of course you're not going to be that good in this regard, and maybe your time to do analysis will be wasted, and then you cannot focus on different experiments. So it doesn't make that much sense. And I'm really looking forward where science will be more like specialized. You have specialists that work together, and not like one person doing all the crazy stuff like PhD students.
Yeah, definitely. I mean, with this increasing, it's kind of an exponential increase of technology evolving. So everything becomes more complex, and it's impossible to do everything at once if you at the same time want to still be in this game of publishing, because of course you can dedicate a lot of time to learn specific analyses, and it's good to understand the basics behind it at least, but otherwise you need to stay still in this sort of race. So I think we need to focus much more on collaboration.
Exactly.
In the future, and it's okay if not everyone knows everything. I mean, that's why also the author lists become bigger and bigger.
Yes, collaboration, transatlantic collaborations, and work in the US. We're going to touch upon this topic in a couple of minutes. Before we go there are two more things that I'd like to discuss with you, and one is the role of microglia in neurodegeneration. Right? I think you've worked in several papers on these things, in particular Alzheimer. Can you tell us more about the role of these cells in neurodegeneration?
Yeah, sure. I can try. So first, neurodegeneration is basically a progressive loss of function or structure of neurons, and that's then driving cellular atrophy and cellular death, and then leads to cognitive decline in then different neurodegenerative contexts like MS, which is more neuroinflammatory, or Alzheimer's disease or Parkinson's disease, which develop over decades, over a long time. Microglia in Alzheimer's disease, I haven't specifically worked on that. Like what we were mostly looking at was microglia from aged human subjects, and when most of the donors that we looked at with the single cell RNA technology come from different neurodegenerative backgrounds, and in order to not have an underpowered study, there were many different diseases included, and the idea was more to have a general framework of how microglia look like in aged humans, and then we try to associate specific subtypes that we describe with specific traits of disease. These are not really statistically super significant data, because the study was underpowered in the sense that we had enough donors from one specific neurodegenerative disease in order to really causally describe phenotypes that are really associated specifically with AD or others. But there is a beautiful paper that I want to mention, which is like 1.5 years old, from the Callis lab. And what they did is a huge study of single nucleus RNA sequencing in Alzheimer's disease, and they described dynamic states of microglia across progression of disease. So what they did is basically look into many different, I think 400 something samples from patients with Alzheimer's disease with different disease states as well. So you characterize Alzheimer's into Braak stages, from 1 to 4, I believe. And they were able to say to describe that microglia go from this homeostatic state, which is the general state, which is characterized by specific marker genes that are expressed on the cell surface, through different stages, which are like an early response microglia state, which has a different transcriptional pattern, and then they go more into an interferon related stage where you have a lot of interferon signaling, and then they go further into a pro-inflammatory stage, which is the one that we already talked about, which is characterized by interleukin 1 beta and TNF alpha and interferon signaling. And in the end, they go into an atrophic state, which is again characterized by specific marker genes. And they also were able to relate specific states of microglia that shift through the spectrum with tau or with amyloid plaques. Mm-hmm. So they kind of also are maybe able to causally or to define which state might be associated with the tau tangles versus the amyloid plaques, which are these two pathological hallmarks in AD. Mm-hmm. And we are also still trying to understand which comes first and how do microglia change based on what is there. Mm-hmm. And more general, there's this disease-associated microglia, which are quite famous, which were described 8 years ago by now in mice first, and then also in humans gradually. And we have also seen these disease-associated microglia. They arise in two different states. They are called DAM1 and DAM2, but that's defined in mice. That's a little bit like the M1, M2. It's mostly more complex than that, and we don't find all the transcriptional signature from mice in humans. But we know that they are definitely associated with disease. Mm-hmm. And one paper of mine tried to and managed to replicate this disease-associated state in a dish in vitro mm-hmm. by specific compounds that we expose the cells to, and then they really drive their state into this disease-associated phenotype. So mm-hmm. now we are able to also functionally study these cells because we don't really know yet whether they are beneficial or detrimental to disease, and also depends on the state again.
Mm-hmm. When you say we don't know what comes first, A beta tau or microglia, you mean what causes this kind of, let's say, dying of neurons, the activation of microglia, which leads to A beta and tau accumulation, or the accumulation of A beta and tau causes the activation? This is what you meant?
Yeah, that's what we don't really know. I mean, there are some studies, and one is also from our lab that tries to kind of associate or infer computationally based on what's known, what might come first, but then to experimentally prove this, and then not in mice, but in humans, obviously is impossible. Mm-hmm. So for now we are stuck with the computational analysis and then validation in human model systems.
Yeah, that is true. It's very hard to study anything neuroscience related in humans just because of these reasons. And interesting in these diseases is that, let's say in Parkinson, Alzheimer, all of the neurodegeneration that is going on, it is actually starting years before the any kind of visible effect is in these humans, or so this is the challenge. Maybe if we can catch it early on and then somehow apply some treatment, maybe we could kind of stop the progression, but we still cannot do that. And it would be cool also to see in the case of microglia if some kind of alteration also happens way earlier, before the onset of the disease even, or not. But I guess this will be very, very challenging, because you don't need to have a biopsy of your brain if there is no symptom, right? So I'm also not aware of some non-invasive imaging techniques that are studying microglia. I don't think there are. Do you know anything for that? About that?
No, I mean, there are PET. I mean, it's possible. So that's another field or other discussion to actually go into. But it might be possible to look into microglia using PET, so positron electron tomography. So that's a sort of contrast-based imaging technology. But currently, the tracers that are developed and that they claim that they look into microglia, they're not very specific to microglia. So they also actually label some sort of a lot of endothelial cells, for example, and I think some astrocytes to some extent. So they are not very specific. As soon as we have novel protein-based microglial markers, which one of my other projects aim to actually describe and find, we might be able to look into microglia more specifically across also trajectories of disease. Yeah, and one more thing that I found really interesting that I have no paper to back up right now, but is that apparently also some brains from healthy donors, so to say, they show amyloid plaques, and apparently also some people that have cognitive decline and that have diagnosed Alzheimer's also show sometimes not necessarily all the hallmarks, the histological hallmarks that we have defined. So also there, it's not that black and white, so to say.
Yeah. This field is crazy. I mean, they're still debating what is coming first. Is this the cause or the effect? And also, like as you said, there are. I'm also familiar with a study. I think of like autopsy of the brains of nuns somewhere in France. I don't remember, where they were like in their 80s, late 80s, 90s, and then there were everywhere signs basically of Alzheimer and all the plaques, etc. But they had no sign of cognitive decline whatsoever. They lived like completely independent and happy and healthy life. So it's very strange. In some people, this is causing huge problems. In others, it's just not there. Some neuroprotection is going on that we probably don't know.
Yeah, it's super interesting. There are specific ethnicities or populations that are actually more resilient to Alzheimer's versus others that are not. And also something we have to keep in mind, which is kind of shown as well, but we don't really talk about it too much, is the exposome. So basically everything, like the combination of everything that we are exposed to in human life, that also impacts heavily how we and whether we develop a disease in one or the other way. So yeah, we can still also we need to find out what it actually does and what exposomes then lead to or accelerate maybe AD development or so. But that's also something to look at.
Yeah, not only AD, also other neurodegeneration. You know, I recently participated in an event where I had to give a talk about our technologies, and it was related also to Parkinson's disease. And when I prepared myself for the topic, and I realized, one of the risk factors for developing Parkinson's disease is actually drinking water from a well, or like work in agriculture, which is like completely interesting, you know. So there is something related to the disease which is outside in this exposome field, you know, and very, very, very strange and interesting. But there is very little research on that, right? Verena, so we can talk about microglia for another hour and so, even longer. But I'd like to ask you two more things before we finish the podcast. Let's start with the one that is kind of you already actually touched upon it, about the collaboration. So you, after your PhD, you went to the US in Columbia University in New York, very, very prestigious institution, where you worked up until basically a week ago or something, month ago, I don't know exactly, but very recently. And I think you are very familiar now with current events that are going on in the US. And can you maybe introduce what is happening now with all these governmental interventions in terms of like cutting funding, how to say, not banning, but like stopping visas of international students, and like basically killing international collaborations in the US? Can you tell us something from the kitchen, for example?
Yeah, I mean, since the beginning of the Trump administration, things have really changed tremendously. And the first few months, like 2 or 3 months, I was still at Columbia. So the first big thing that the Trump administration did was to cut the indirect costs from 65% down to 15%, which is a tremendous cut. Potentially a cut that was a little bit expected, but not in this dimension.
So just to interrupt you, what are these indirect costs covering? Do you know that?
So indirect costs are basically, if you imagine you get a grant as a researcher at a university, you get $1 million, for example. For every dollar that you get, with the old indirect costs, the institution got 65 cents per dollar on top to maintain facilities, to pay the rent, to pay electricity, to pay core facilities and cleaning and just for maintenance. So by cutting down from 65 cent per dollar to 15 cent per dollar, there's a massive hole, obviously, in the budgets of all the universities, which was one of the big problems or one of the big cuts that started first to happen, which also led, of course, to a lot of consequences at universities. So they had to react quite fast. Certain things are now more difficult to do, going to specific conferences only on funding money, or hiring stops occurred across the country in specific universities. Maintenance most likely will somehow decrease at universities because their budgets are much lower, and only clinical research staff was allowed to be hired for a certain amount of time. Not sure if this is still the case. And then on top of that, there were these rather targeted actions of the Trump administration to specifically cut funding in universities that were big in protests against the Gaza war, the war in Gaza. So now a lot of different universities are affected by that. There are, I think, at some point there were 60 universities on this list, and they were kind of targeted with like funding being withdrawn. Then some universities fought back, others did not so much. And right now the most prevalent case is Harvard, where the administration fought back, and where now I think it's at the Supreme Court, and there was a stop, so to say, of what the administration imposed onto Harvard right now. So that's where we are right now. I also read that for 2025, the Trump administration cut the budget for funding and research, for research funding, down from $2 billion onto $980 million. So it's cut down by 51% or so.
This is the NIH funding or National Institute of Health, right? This is one of the main funding agency of science, right?
Yeah. So that's the overall funding that is cut down. That's like information from the National Science Foundation, NSF. That's what I read this morning. So of course this then trickles down into many different things and accelerates crises that we already have, like the postdoc crisis, and obviously people that are in specific grants that might be affected might not be able to stay longer.
That is crazy. It's so ridiculous that you have like so many top-notch institutes that are super internationally prestigious, very well connected and established, and now basically you kill this with 2-3 different measures. And then I don't know, how is the feeling? I heard about from several places, like people from the US, they look for opportunities now in Europe, to go there, come here, actually start working. And like the working environment is very like close. You don't share information that openly. It's very, very bad, actually. I don't know what to say. And I wanted to go to the US, for my postdoc, but I don't think that now is the right time to do so. I don't even know if I will be allowed to. They don't hire. I mean, there is no option to go there, I guess. So
yeah, right now it's pretty much impossible. It's kind of expected that there will be a huge brain drain or a huge migration of scientists to other parts of the world.
But hey, I mean, there is always brain drain to the US, so now science there will basically suffer, and at some point it's when the wind blows the other direction, then people again will return or go there. So yeah, it's how life goes on. Well, unfortunately, things are like that, and in Europe doesn't seem better, I must say. Like as long as I'm familiar, for example, Free University in Berlin, they also have like some 40 million cut in research and stuff like this, and this is not the only one. So now with all the policies going on in Europe for militarization and whatever, but this is a different topic. Let's go to some other initiative that you started a while ago. It's called SciLead, and it's organization that... what is SciLead?
SciLead stands for Science Leadership, and it's an initiative or an NGO that we means me and 3 or 4 others. We founded this 4.5 years ago. So it's based in Berlin, and it's basically an initiative that is grassroots based or grassroots movement initiative that advocates for more leadership and mentorship in academia. So when we met in the realm of a meta research conference 4 or 5 years ago, and we were all a little bit we all experienced some difficulties in mentorship and leadership throughout our academic careers, and we decided that we need to advocate for that. So what we did first is actually a big survey across Germany to look into the leadership state at academic institutions in Germany, and we found that most people, so leadership for us or mentorship doesn't only start when you are PI. It starts very early on, because sometimes you already have to supervise some bachelor student when you're still in your master's, or when you're during your PhD, you already supervise students, and then it increases gradually. But leadership starts very early. So what we found is that most people, and that's true for PhD students to professors, are not really, they don't feel ready for being in a leadership position. But how the current system works is that we get promoted based on publications, based on grant funding, based on citations into positions where we have to lead, but we are not really equipped with the skills to actually do so. So this initiative is advocating for more of that, and we offer a lot of workshops, we do community building events, and they are all free, and we just try to build awareness gradually in different dimensions and different target areas, targeting different people to become more aware of the topic. Meanwhile, also many institutions have leadership trainings when you enter as a postdoc or when you enter as a junior PI or something. So you get some specific training. But what we believe is that this should be accessible maybe already during your studies in your master's or so, because you most likely aim to be somehow in a leading position if you go for this sort of education.
I mean, leadership is very valuable, not only in education, academics, but also in business. And if you want to start a company or in any domain, basically, of life, it's very important to have these skills. Of course, not everybody has to be a leader. There are people with different qualities and characters, but for those who want to, then it's very, very important to engage in such activities and participate in such workshops, events. When you have some of these workshops, are they in person or are they online, or is it mixed? How is it going?
It's mixed. It's been mostly online. So we are right now across Europe and Mexico. And before that, we were in the US and in Europe. So most events are online, so we can also reach more people. We have actually quite audience from different countries present usually, which is beautiful. But we also have in-person events. So we are actually now just starting some sort of small collaboration with the TU in Berlin for an open science sort of initiative that goes through the Berlin University Alliance. And so we aim to actually give like an in-person workshop there, for example, to also reach more locally people. Yeah.
That's cool. Maybe you can come by to Göttingen, to give some seminar here, or there are these biannual student organized conferences. One is Neuroscience. I mean, it's the only one that's biannual, and Encephalon, I think, is also biannual. No, not anymore. There you have Horizons. These are all like conferences organized by students, by PhD students, and they always have like one day like career fair, building your skills. Maybe you guys can come at some point and spread the word here also in Göttingen. We have a lot of scientists. Yeah, sure. I would be happy to give you more information off the camera. I think this is a good topic to end our discussion and our podcast. Verena, thank you very much once again for coming and for sharing all of these interesting insights about microglia. And I'm sure that you're gonna show the world also many more cool stuff, not only about microglia.
Thanks very much for the invitation, SciLead.